So I wanted to demonstrate what we've been able to accomplish since we opened the Institute with respect to some examples of the translational research that we've been able to do. And just very quickly, since we're now in one facility, although it's somewhat virtual, we have the clinic together with the research, and I meet at least once a week with the director of research. So it allows things to happen very, very quickly. And I'll show you the projects we've done in about the last year and a half on a relatively limited budget. And I think it's a model that can be used elsewhere for rapid research and turnaround here. And the projects that we've been working on, I told you the DNA where we did not find a clear-cut single marker, gene expression, virus expression, clonal cell T rearrangements, and chromosomally integrated HHV6. And we already know, I wanted to briefly show you, in order to support Jonathan's work, what the CDC has produced with respect to their gene expression profiling. And the technique, you already know about it. It's interesting that they used resting specimens rather than activated, rather than after exercise or after some kind of provocation, because one wouldn't necessarily expect to see the changes in CFS patients at rest. But that's how they perform this. And as Jonathan has told you, they did not confirm their findings, or they have not yet in any case. And studies have been somewhat mixed, and people have different footprints that they think they would like to use. This is their famous slide where they took 50 symptoms and 39 genes and put them in a model and said you can clearly differentiate true chronic fatigue patients from non-fatigued. And this group in the middle that the CDC insists on continuing to define is patients who by symptoms don't meet the criteria. They don't have sufficient number of criteria. And this is one of their famous slides here that I think is actually very interesting. It's the details of it don't matter, but you see on the left-hand side with the red being down-regulated genes and the green being up-regulated, and they could differentiate acute onset, which is on the left, from chronic, slow, progressive chronic fatigue on the right. I think that is very dramatic, saying probably, even on a genetic level, these are two very different processes, at least as we measure them by messenger RNA. On the right is a nice summary that, and it absolutely confirms what Jonathan said earlier, in that you see some rather striking differences between the patients and controls with respect to so receptor genes, metabolic genes, and transcriptional genes, and of course the list goes on and on and on, because I think they looked at a very selective array of, I think, 1,600 genes or something like that. So it wasn't nearly as thorough as other researchers are doing. And this is interesting. I just put it up to show that they took patients with acute Epstein-Barr virus infection and studied them after 1 year to see if there was a difference between the ones who recovered and the ones who stayed sick. And in fact, the blue-purple genes are the ones that were present after 1 year in the patients who stayed sick, and the lighter colored are the ones who recovered. So there's a very distinct difference in the gene profile at 1 year in patients with the acute onset of EB and the ones who stayed sick and the ones who recovered, which is very provocative, actually. And then, of course, their sort of dynamite slide here that they're using to differentiate normals based on just 4 genes versus chronic fatigue patients, and further saying that with 2 specific genes that you see on the right, they can subdivide the patients into various categories depending on duration of illness and things like that. There's obviously an enormous power to this if this can be replicated by other sites, and would make it actually fairly simple to, if it holds up and is specific for chronic fatigue. The controls, again, have been normal controls. We're doing this with rheumatoid arthritis, hepatitis C, HIV, the same kinds of things that Jonathan has said for controls. Now, I'm particularly interested in a group of patients that I found that had developed, in longitudinal studies that we've done over 20 years, we find 2 neoplastic disorders that have occurred in much higher than expected frequency. And one is brain tumors, and the second is lymphomas. And what was most interesting about the lymphomas is the vast majority of them have all been mantle cell lymphoma, which is a very unusual lymphoma. It's very aggressive, and I currently have 7 cases, when statistically that would be chance in my practice is 30 million to 1. So there's clearly some correlation here. And what I started looking at these patients, and much to my surprise, we found a gamma receptor clonal rearrangement of the T cells on 17 of 50 patients who were screened, who had what I felt clinically would be group B symptoms. They had adenopathy, weight loss, night sweats, etc. So I had a high index of suspicion that they might fit into this category. And so we thought this would be a great group to look first for the viral array to see if there was some kind of viral origin to this disease. And the reasons for this, of course, is that herpes virus has been associated with lymphoma before, and we were very concerned about this group that had developed mantle cell lymphoma, since it's so aggressive, and the etiology of mantle cell lymphoma has really never been determined. The genetic marker, the cyclin D defect, has been described for a number of years, and these patients have that particular defect as well. So our hypothesis was that perhaps a co-infection of unknown viruses, or 6A or EB, or some combination of the herpes viruses, might be leading to this gamma T cell rearrangement, and that perhaps we could be proactive here in the prevention of lymphoma in these patients. And we used a mammalian virus expression microarray to look at this, and we took 3 groups of patients. We took the patients who had developed mantle cell lymphoma, the patients who had clonal T cell rearrangements but did not have mantle cell lymphoma, and then we took some, quotes, normal chronic fatigue patients. And here's the technique that was used, and for those who are technically interested, I can give you the details later. But it was a 10,000 feature probe that's used at the National Cancer Institute in their cancer research for the relationship between viruses and cancer. And each virus is represented by at least 10 sequences, and both RNA and DNA viruses are tested for. They allege that all known viruses that can affect humans and birds are tested in this particular array. So it's really quite a powerful array. I was not prepared for the results that I found, however. We found 1,608 viral transcripts, which is a huge amount in the patient population. We're sorting our way through that because, as Jonathan says, this has to be confirmed by PCR, and that's a somewhat tedious task to do, but we're in the process of doing that. And we took the top 300 to 600 transcripts, and we're looking at 50 of those viruses. I can say on our preliminary analysis of this at this time that the herpes viruses definitely predominate in the group that have T cell rearrangements and in the mantle cell lymphoma group, and much less in the control group and in between in the chronic fatigue group. There are some unusual findings. There's some suggestion of a either mutant or new herpes viruses that came up in this particular array, but again, we have to confirm that. In the normal patients, we found human endogenous retroviruses, rhinoviruses, adenovirus, and you'd expect that in a control group, I think. To answer some of those questions, we're repeating the microarray now, which should erase the adenoviruses and the rhinoviruses and the situational ones that appeared at that point in time. So we're doing the same patients over again. We are in the process of doing the PCRs, as I mentioned, to confirm this, and we're beginning collaboration with other centers, because the power, of course, will be to demonstrate the same thing in other population groups if it's going to be a useful technique. And it appears that, in fact, we have some viruses in common with those that have preliminarily been reported from here. Another area of interest that we took on as a project over the past year has been the germline chromosomal integration of HHV6. As many of you know, there was HHV6 variant A was first isolated from Lake Tahoe patients and subsequently characterized at the NIH. So that has been a virus that I've been very interested in over the years, because it's neurotrophic and lymphotrophic and can cause has been associated with some disease processes, in contrast to HHV6 variant B that is associated with childhood rash and acute febrile illness. So HHV6 is unique in that it can chromosomally integrate. It has an integrase gene that's easily identifiable, and we even know where it inserts because we have done sequencing on this particular topic. And the object of the study was to see the prevalence of chromosomally integrated HHV6 in American patients, again using the same groups that I told you about before, the T-cell rearranged group, the lymphoma group, and the normal, quotes, chronic fatigue patients. And the reason for this is that the Japanese reported an incidence of chromosomally integrated HHV6 of about 3%. But that's variant B because they don't have variant A in Japan, so, or at least not reported. So this was well, here's again the technique that we used in order to do this. This was done on hair follicles. You could use nails or you could use other cells when you're looking for chromosomal integration. Very specific test. It was quite accurate in our controls here. The summary is just to point out the fact that when you look at it, we found about a 3% rate of chromosomal integration in the population at large. I stumbled actually upon a family, and I want to stop. This is very interesting to me because two of the children are severely ill with chronic fatigue and have evidence of HHV6 reactivation and are, in fact, chromosomally integrated. 3 of the children are chromosomally integrated, and the father is as well, although the father is alleged to be asymptomatic. So this is extremely provocative for those people who are interested in hereditary passage of herpes viruses and chromosomal integration. It's a big topic that's somewhat peripheral to the mainstream of chronic fatigue syndrome, but I think is really interesting and perhaps critical for the future. So in summary, we did not find an association with chromosomal integration and the lymphomas. So it was a null hypothesis on that. However, we did confirm a fairly high rate of chromosomal integration of about 3.3%, which was higher than was predicted. 2 of the 3 patients that have clonal T-cell rearrangements did, in fact, have chromosomal integration, and what that means remains to be determined. For those of you who aren't familiar with gamma T rearrangements, it's reported in CMV infection but resolves with resolution of the infection. It's been reported generally as a pre-lymphomatous marker, which is why I'm so concerned about it. And that's what we've accomplished in our translational research model in the past 18 months.